Welcome. Today, we're digging into the fascinating world of urban archaeobotany. This field offers a unique window into the daily lives and surroundings of ancient city dwellers. It's all about recovering and analyzing plant remains from archaeological sites to reconstruct what people ate, how they managed their environment, and even gain insights into trade, social status, and rituals. You might also hear it called paleoethnobotany. So, what do these plant remains tell us? Evidence comes from two main types: macrobotanicals and microbotanicals. Macrobotanicals are things visible to the naked eye: seeds, fruits, wood, charcoal, nuts. These are invaluable for understanding diet, showing what staples were eaten or if exotic fruits suggest trade. They also reveal agriculture, like what crops were grown or what trees were used for fuel, helping reconstruct local woodlands. We even find charred food bits, direct evidence of cooking. Then there are microbotanicals, requiring specialized techniques. Pollen analysis, or palynology, reconstructs past vegetation and even climate, showing deforestation or new plant introductions. Phytoliths, tiny silica bodies from plant cells, help identify plants like grasses and can be found in sediments or even on tools and teeth, directly indicating consumption. Starch grains, found on pottery or tools, can identify specific plants that were processed or cooked. How do these delicate remains survive and how do we find them? Preservation is key. Charring from fires makes plants resistant to decay. Waterlogged sites, lacking oxygen, can preserve even delicate items. Dry, arid environments also naturally preserve organics through desiccation. Sometimes, minerals replace plant matter, preserving its form. To recover these, flotation is a major technique. Soil samples are added to water, and charred remains float up. Sieving and soil coring for pollen are also used. For analysis, microscopy is crucial. Stable isotope analysis on bones and plants details diet. Ancient DNA can identify species and trace crop history, and residue analysis on pottery reveals specific processed plants. What does all this tell us about ancient urban life? We can distinguish between local and imported resources, mapping trade networks. We see the environmental impact of cities, like deforestation or the creation of gardens. An increase in date palm pollen, for example, often signals human activity. Differences in plant remains across a city can suggest social status, like variations in diet between elite and workers' areas. And it's not just about food. Plants were used for rituals, medicine, textiles, fuel, and construction. The field is always evolving. There's more interdisciplinary work, combining plant data with animal bones and artifacts. Advanced imaging and chemical analyses offer deeper insights. Big data and shared databases are enabling broader studies. There's a growing interest beyond just food, exploring all the ways plants were used. Ultimately, urban archaeobotany helps us understand how ancient urban societies adapted to and modified their environments, offering lessons on human resilience. By piecing together this botanical puzzle, we get a richer picture of complex ancient urban life and our ancestors' connection to the plant world.